Our last speaker for the oncology section is Dr. Shilpa Gupta. She is a hematologist-oncologist in Department of Hem-Onc Transplant here at the University of Minnesota. She was recently promoted to associate professor, so congratulations again.
Thank you.
She is a practicing genitourinary oncologist and has had involvement in over 25 clinical trials as a principal investigator. These are phase 1 through phase 3s. She's had extensive experience working with targeted agents as well as the immunotherapies. Now, these immunotherapies are a newer treatment entity, and we've heard lots about the effectiveness of these agents in patients who had not previously responded. However, they don't work in everybody, and so I think Dr. Gupta can speak to maybe the importance of identifying the need to identify markers that can help us better predict which patients respond to therapy. For example, she's had experience working in the Keynote 12 phase 1 trial, which is a study of Pembrolizumab, and so she's had extensive experience working with these immunotherapies. So, Dr. Gupta?
Thank you. Thank you, Mark, for the introduction, and I would like to thank the organizers for inviting me to speak. Thanks, David, for leading, providing a nice segue for my talk. It makes my talk a little easier. So these are my disclosures, and as all of you know, immunotherapy has become an integral part of how we treat cancer nowadays, and these were the updated hallmarks of cancer which incorporated immunotherapy, because there's various mechanisms that immune system plays in the treatment of cancer. For example, evading immune destruction, dysregulating cellular energetics, genome instability and mutation, and tumor promoting inflammation. And immunotherapy will be the future of how we treat cancer in addition to targeted therapies. For the purposes of this presentation, we'll focus on checkpoint inhibitors because these are the agents most far ahead and have received a variety of approvals across cancer types. And the CTLA-4 and the PD-1 immune checkpoints are negative regulators of T-cell immune function. And as you can see, this is the mechanism of action by which they unleash the immune system against cancer, releasing the brakes that occur. And this is the immune checkpoint inhibitor landscape. As you can see, within the last 5 to 6 years, we've made tremendous progress. And actually, my slide is already out of date because just last week we received approval for 2 gynonc cancers, endometrial cancer and cervical cancer, for checkpoint inhibitors. But you can see the impact this is having across tumor types, no matter what immunotherapy we are talking about. Pembrolizumab and nivolumab are the most widely approved ones, but in bladder cancer and lung cancer, we also have others like atezolizumab, and in Merkel cell cancer, we have avelumab, and in bladder cancer, we also have 3 other immunotherapies, durvalumab, as well as pembrolizumab, nivolumab, atezolizumab. So there is a need for precision oncology in the immunotherapy era, because checkpoint inhibitors are currently used to treat multiple types of cancers with notable success. And in the refractory setting and first-line non-small cell lung cancer, they've really changed the way we treat cancers, whether it's lung cancer, kidney cancer, bladder cancer, lymphoma, or any other cancers. However, as you can see in this slide, the benefit is limited to a fraction of patients. Not everybody who gets checkpoint inhibitors derives a benefit. For example, in bladder cancer, only about 1/4 of patients actually respond to immunotherapies. And across cancer types, there is no cancer where 100% patients will respond to any given immunotherapy. Therefore, it is very important for us to identify predictive biomarkers of response to optimize treatment selection and also provide cost-effective treatment, because these drugs have a lot of financial toxicity, as you all are aware. So this is another slide which is highlighting the importance of the majority of patients not having a response to PD-1 blockade. And this is across these different kind of cancers. and just underscoring the point that we need to develop predictive biomarkers. And there have been several recent negative phase 3 trials, including the nivolumab trial in lung cancer, the MYSTIC trial in lung cancer, IMvigor 211 with atezolizumab in bladder cancer, and IDO inhibitor trials. And we need to learn from these trials and design better trials based on useful markers so we can select patients appropriately. Now this is a graph showing the PD-L1 expression and clinical benefit across cancers. As you can see, there's some hint here that PD-L1 expression may imply that patients would respond better compared to those who do not express PD-L1. However, those who do not express PD-L1 also have modest responses and sometimes better compared to chemotherapy, and that is why they have received approval. And most of the cancers do not require PD-L1 testing except for first-line lung cancer, where FDA has mandated checking PD-L1 for use of pembrolizumab, because benefit was seen only in patients with more than 50% PD-L1 expression. And this is the study I was alluding to, the Keynote 024, which is pembrolizumab first line in non-small cell lung cancer. And in this, the bar was set too high. It was PD-L1 expression 50% or higher. And you can see the dramatic separation of curves, both for progression-free survival and overall survival. And this is currently approved in this setting. Development of effective biomarkers is not an easy thing to do, as we've heard this morning. We need to have a good assay, validate that assay, then develop clinical validation, clinical utility, and then generate companion diagnostics, which by definition are essential for safe and/or effective use of therapy, and complementary diagnostics, which may help us to decide the risk and benefit of decision making. Now, the field is very complicated because we have multiple checkpoint inhibitors, which is great, but each checkpoint inhibitor uses a different assay and a different platform for the PD-L1 testing. For example, if you look at nivolumab, which targets PD-1, the antibody clone is 288, which is a DACO platform, and the tumor cells are evaluated for expression of PD-L1. But pembrolizumab, which is also an anti-PD-1 antibody, uses another antibody clone called 22C3, and in this, the tumor cells or immune cells are evaluated for PD-L1 expression. Atezolizumab, which is another antibody, it's anti-PD-L1 antibody, it uses SP142, and there's a whole different thresholds for scoring, and so is for durvalumab. So it's not that you want to give a patient a checkpoint inhibitor and you can just order PD-L1 testing. You have to order a specific PD-L1 test for the specific antibody. So it's not as simple as it seems. It does lead to a lot of confusion. And independent and unique test for each antibody has different platforms, different scoring, different interpretation guidelines, and multiple companion and complementary diagnostic pairs are entering the market. So it leads to complex, challenging decision-making problems, both for the pathologists as well as clinicians. And running a different test for each drug is impractical and a waste of tissue and money. And running one test for all drugs is also not ideal. So there needs to be a standardization for this, and that is why the Blueprint project was launched, which is a really remarkable initiative, and the goal of this project was to agree and deliver via cross-country collaboration a package of information upon which analytic comparison of various assays may be conducted. And this is the first time several pharmaceutical companies and academic institutions have come together to lead to this standardization to make the field easier moving forward. And this is the data that was published last year from phase 1 of this Blueprint project. And in this you can see that most of the antibodies are actually quite concordant, except for the one in light blue, which is the SP142 for atezolizumab. And this was again another initiative across 90 archival lung cancer tissues across multiple institutions. And here also they found that SP142 is the outlier. So while the other antibodies were falling within the same range, the SP142 seems to be an outlier, and it seems to be the odd one out. And I want to just highlight this trial because SP142 is designed for atezolizumab, and this is a key example of a trial that can be negative if not planned properly. So atezolizumab was actually the first immunotherapy to be approved for bladder cancer in 2016 based on phase 2 trial. And then when they did the phase 3 trial, IMvigor211, randomizing atezolizumab versus investigator's choice chemotherapy in refractory bladder cancer, they stratified based on the PD-L1 testing, because in the phase 2 study there was a hint that higher PD-L1 expression leads to better responses. And unfortunately, this trial was a flat-out negative trial. It did not show any difference of benefit between PD-L1 positive or PD-L1 negative patients. And we don't know if that is because of the assay that may be at fault or what are the other factors. We're still trying to figure that out. So these are the curves that you see. The PD-L1 high expressors, patients did not have any difference whether they got atezolizumab or chemotherapy. They performed similarly. And those with PD-L1 low scores, the atezolizumab patients actually did somewhat better than chemotherapy. So that's the opposite of what is expected or what is believed to be the theory. So we're still learning what that means. So the take-home messages for PD-L1 testing is that PD-L1 testing by immunohistochemistry can guide treatment decisions in some cancers, like first-line non-small cell lung cancer, where you require a 50% or higher PD-L1 score for prescribing pembrolizumab. However, recently pembrolizumab and chemotherapy combination was approved in the same setting, and that does not require the 50% cutoff. However, as I just pointed out, PD-L1 is an imperfect biomarker, and we need better biomarkers to guide patient selection for immunotherapy. Now I'll talk a little bit about microsatellite instability as a predictor of benefit. David already presented you the basics and how the first time FDA actually approved a drug based on a target and not across tumor types. So DNA repair defects expose cancer cells to immune system, and that is the rationale that why these tumors may be more susceptible to immunotherapy. And this was the original article that was published in 2015 showing PD-1 blockade in tumors with mismatch repair deficiency. These were both colorectal tumors as well as non-colorectal tumors, and patients with the mismatch repair deficiency did better with pembrolizumab. And again, these are the curves showing improvement in progression-free survival as well as overall survival in those with microsatellite instability. And in May 2017, FDA approved pembrolizumab for MSI-high and MSI-high cancers regardless of the origin. And this was the first agnostic indication. And this was based on the demonstration of durable responses in around 150 patients across 5 different trials across tumor types. Out of these, 90 patients were colorectal cancer, and remaining 59 patients had other cancers. And this is, like I said, we can't highlight enough that the FDA has really taken a stand with advancing the field instead of trying to do multiple trials using the same biomarker. Now talking about nivolumab in patients with metastatic DNA mismatch repair deficient or microsatellite instability high colorectal cancer. This was a phase 2 trial where patients with microsatellite instability are randomized to nivolumab versus nivolumab and ipilimumab. And ipilimumab is an anti-CTLA-4 antibody. And FDA has granted accelerated approval for nivolumab in this setting last year. And furthermore, the data from this year's update showed that patients who got nivolumab and ipilimumab had much higher responses than compared to nivolumab alone. So FDA has actually put it as a breakthrough designation, and this combination may get approved down the line. And now I'll jump gears to talk about tumor mutational burden as a predictor of benefit from immunotherapy. And this is a very upcoming field, and we hear about it all the time. Anytime we order next generation sequencing, this is reported. So we need to really understand what this means and what it means for the prescribing treatment. So tumor mutational burden is an independent predictor of response to immunotherapy across a variety of cancers. And as you can see, the differences between the red and the blue dots, patients showing responses have higher tumor mutational burden. Patients showing disease progression or stable disease have lower tumor mutational burden across cancers. And this was presented at ASCO this year, that association of high tumor mutational burden and atezolizumab efficacy across multiple tumor types. And I'll just highlight some of the key slides from this presentation. We know there's the hypothesis that tumor mutational burden is a candidate predictive biomarker of immunotherapy benefit. And the predicted neoantigen load is a component of tumor mutational burden that has been linked to immune response. And this is in the IMvigor study, that is atezolizumab in bladder cancer. They did find that higher tumor mutational burden did correlate with better responses. However, no one knows what is the definition of high TMB. You know, it's reported in this study that 16 or more mutations were considered high. However, in studies for nivolumab, 12 was considered high. So we really need to, again, have this solution of standardizing tumor mutational burden definition as opposed to defining a different cutoff based on different agent and different method of next generation sequencing used. So this is again the biomarker update study from the IMvigor 211, which was a negative study in terms of clinical efficacy of atezolizumab versus chemotherapy. But we learned a lot from the biomarker data that was presented recently. So this is the trial design I already showed you. The trial was negative because patients with high PD-L1 did not do better with atezolizumab. On the other hand, patients with low PD-L1 actually derived benefit. So we already showed that the PD-L1 was not a good selection for this study. But in this study also they looked at tumor mutational burden, and urothelial cancers generally have high mutational burdens that can contribute to increased immunogenicity. And in this study they showed that PD-L1 expression does not correlate with tumor mutational burden when tested with Foundation 1. And in this again, they showed that these are two different entities, the PD-L1 and tumor mutational burden. So you have to put everything in perspective and have more data from trials to actually know which is a better biomarker. They did see that overall survival was better in patients who had high tumor mutational burden, and patients who had low tumor mutational burden, there was no difference between atezolizumab versus chemotherapy arm. Now looking at overall survival based on tumor mutational burden and the PD-L1 status, TMB high patients derived a benefit with immunotherapy. High PD-L1 patients derived some benefit in this, but TMB high and high PD-L1 derived the maximum benefit. But what are the logistical challenges of TMB analysis? The problem is again the same problem we face with the PD-L1 biomarker. There is no standardization for calculation and reporting. Different tests may report different measurements, and there's no one way of calculating TMB. We need to develop a standardized way of calculating and reporting it. And the good news is that there is a TMB harmonization working group that has been formed with the goal of standardizing the definition of TMB and establishing an analytical validation approach and ensuring optimal clinical implementation. And again, several companies, the diagnostic companies, are seeking agreement on analytical and clinical validation studies to support a standardized method of TMB reporting. And the 3 steps consist of in silico analysis, in which the publicly available TCGA data is analyzed to identify sources of variability between TMB calculated using whole exome sequencing and targeted panels in the clinic. And then in fall of 2018, we are expected to come to stage 2, where the cells derived from human tumors will be studied, and they'll agree upon creation of a universal reference standard using whole exome sequencing. And finally, sometime next year, we should be able to get data from retrospective analysis using patient outcome data to assess the variability and define a clinically meaningful cutoff of TMB, so that it can guide clinicians at the point of care and help in molecular tumor boards and precision medicine. And lastly, I'll just mention very briefly, this is also another marker that has been reported in some studies, gene expression signatures. It's not really prime time yet, but I'm going to present some data. Again, we'll go to the reference IMvigor 211 study with atezolizumab in bladder cancer. And this was a 3-gene TGE3 signature that was developed as a surrogate for PD-L1 IHC expression. And interferon gamma encodes the interferon gamma, and then RNA sequencing was used, and around 800 patients were evaluable for this in the study. And they found that the overall survival trend was similar in both the PD-L1 testing and the TG3 group. So it could be a surrogate marker for PD-L1 testing. And similar overall survival was seen in the low PD-L1 groups and the TGE3 groups. And high tumor TGE3 status was associated with better prognosis in both arms. And the PD-L1 enriched these patients with higher T effector immunity. And this is another signature interferon gamma-related mRNA profile, which has been reported with Pembrolizumab studies. And the data shows that higher signature does imply better responses. And this was work we did as part of the Keynote phase 1 study in head and neck cancer, where the genomic determinants of response to pembrolizumab in head and neck cancer was found that the gene expression profile was predictive of response. We have to understand that biomarkers are not really static. You know, as cancer evolves, the biomarkers keep evolving. It's a dynamic state, and hence using one tissue from a point of time may not give us the full picture of a patient's disease. And that's the struggle we are having with a lot of patients having resistance to therapies. So the challenges and future directions are that I think while we've made a lot of progress with immunotherapy and we've identified some potential predictive biomarkers, we still don't have that one predictive biomarker like the HER2 or hormone receptor status in breast cancer that can tell us which patient will benefit from immunotherapy. And it's not just PD-L1 or tumor mutational burden or gene signature. It's actually requires integration of multiple biologic components, because immune therapy acts across a variety of immune components in a patient. And gathering information from one piece of precious clinical sample, and many times we don't have tissues available, we really need to use it judiciously to integrate the data into biologically and clinically actionable information in the context of a rapidly changing therapeutic landscape. And we have to make decisions based on what we have available. So we don't want to go on a random hunt, and we really need to get more data before determining which is the best biomarker. So while we have made a lot of progress, we look forward to more exciting progress in the field, and I'll take questions if any.
So I have one question on the tumor mutation burden. So it's based strictly on the number. Is that correct? So do they consider locations of these mutations? Like, is the power too much of a challenge to try to sort that out?
Yeah. To my understanding, it's just the number. And David, well, do you have any thoughts on that? I don't think they do anything else, right? Yeah. And that's why it's not the ideal marker.
Okay, well, seeing no other questions, thank you, Dr. Gupta.
Thank you.